Good morning, friends. Last class we have discussed about the discovery of auxin. Today we will understand the biosynthesis of auxin in plants. To understand the biosynthesis of auxin, we have to first understand the biosynthesis of tryptophan. Tryptophan is an amino acid which is known as the precursor of auxin. So to understand the biosynthesis of tryptophan, we will go to the cyclic acid pathway. In cyclic acid pathway, one biochemical known as chorismate is derived. Chorismate is the one which is starting the process of biosynthesis of this tryptophan. Chorismate is converted to anthranilate by the enzyme anthranilate synthase. Anthranilate again with the help of enzyme for phosphoribosyl anthranilate transferase enzyme converted to 5-phosphoribosyl anthranilate. This product with the help of this intermediate product is converted to 1-O-carboxyphenyl amino 1-deoxyribulose 5-phosphate with the help of this isomerase enzyme. This product is now converted to indole 3 glycerol phosphate, that is IGP, with the help of IGP synthase. Now this IGP is very important as it is producing indole. Indole is produced because of the isomerization of IGP. Indole with the help of serine, an amino acid, is converted to tryptophan. So this tryptophan and indole are two important molecules in terms of biosynthesis of auxin. So indole can be converted to IPA, indole pyruvic acid, and then will produce IAA. And tryptophan also can undergo a number of processes where it is converted to IAA. So there are two pathways where IAA is produced. One is tryptophan independent pathway, another is tryptophan dependent pathway. Tryptophan independent pathway is starting with indole, but tryptophan dependent pathway is starting with the tryptophan. Now we will understand the IAA biosynthesis pathway. So IAA biosynthesis pathway, if we see, there are two important parts. One is tryptophan dependent, another is tryptophan independent. Tryptophan independent, we studied that it is starting with indole, but tryptophan dependent pathway are of dependent pathway are of two types. One is IPA, another is tryptamine, that is TRM. It is written as TRM here. So IPA is the one pathway and another pathway is TRM. TRM is now again is undergoing in two processes. One is indole acetonitrile and another is indole acetamide. Here it is the indole acetonitrile and it is indole acetamide. So there are two pathway again, and this tryptamine pathway is also known as YUCCA pathway, as the gene responsible for this pathway is YUCCA. So if we see this IPA pathway, tryptophan is converted to IPA, IPA is converted to indole acetaldehyde, and this will produce indole acetic acid. But if we see this tryptamine pathway, as I told that there are two ways. It is indole acetamide. So in tryptamine is converted to indole acetal doxime, and indole acetal doxime will convert indole acetamide, and indole acetamide will convert it to indole acetaldehyde, and this indole acetaldehyde, as in IPA pathway, it will convert it to this IAA. Okay, and third pathway, that is the IAN pathway, indole acetonitrile pathway. It is undergoing indole acetamide nitride oxide, and then it is converted to another molecule where there is presence of indole glucosinolate, and this pathway is generally occurring in Brassicaceae family. So now we can brief this class, as there are two pathways for IAA synthesis. One is tryptophan dependent and another is tryptophan independent. Tryptophan independent is starting with indole, and tryptophan dependent is starting with tryptophan. Now tryptophan dependent pathway is having two types or two paths, that is indole pyruvic acid and another is tryptamine. Tryptamine is again having two paths, that is indole acetonitrile and indole acetamide, and all three are undergoing through processes and producing indole acetic acid, that is the IAA. So this is all for today's class. If you want me to discuss about any other particular topic of plant physiology or plant science, let me know in comment section. For more such videos, please like, share and subscribe. Thank you.